Hello everyone, my name is Ashfaq Ahmed and I work in the field of structural biology and bioinformatics. Today, I brought a very interesting tutorial once again. Its name is Performing Phylogenetic Analysis on Nucleotide Data in Colab. It is a complete tutorial where we will learn how to perform professional kind of phylogenetic analysis and how it will help us in our research and thesis or manuscript writing. So let's start. But remember, this tutorial is all about the nucleotide data, to what I can say genes, DNA, genomes, etc., etc. For if you are dealing protein data, or amino acid data, or I would say protein sequences, then for that I will upload another tutorial soon. So let's start. So first of all, as we will cover this tutorial on Colab, which is very easy for most of you guys. So that is why first of all you have to provide this address in your browser. I will also place this link in the description section of this video. Then you have to come here and select alignment. Once the alignment is selected, you will see phylogenetic analysis nucleotide sequences. Now click here, and once you click here, then you have to connect the notebook. Okay, now let me show you the data I will use in today's tutorial. Okay, this is my desktop and I have this directory data, and it has one file. It's a text file rbcl underscore gene dot txt. Okay, so I have all these genes data of almost 19 or 20 genes. We can confirm it from the bioedit. Desktop data rbcl genes. So these are total 20 genes. Okay, now just a quick look uh, for these sequences. These are not aligned, you can see. So this is our data from where this data came. So let me show you. I downloaded this data from the blast. And if you don't know how to download your data from the blast, recently I'm covering introduction to bioinformatics or learn bioinformatics from the scratch. So I would suggest watch that series. You will learn how easily you can download data for your nucleotide or genome sequences. Okay, and okay, oh, I can see it has two sequences with the same name, like this one and this one. So that's okay. I will delete one of them and I will come here, edit and delete sequences. Now we have 19 and I will save it. Okay. Now this was my data. So I just mentioned to you or shown you that this kind of data we will use today. Okay. Okay, a brief introduction about this notebook I prepared. In this notebook, we will use two programs, MAFFT and IQ3. Okay, with the MAFFT, we will also align our dataset. Also, we will generate a guided tree. And with IQ3, we will generate a professional kind of tree. What is the difference between guided and professional tree? We will discuss in detail. Okay. If you are using this Colab notebook for your analysis or thesis and you need some citation. So there is a citation for MAFFT, this is for IQ tree, and this is for model finder. We will learn like what are these and when we will use these citations. Okay, the first step is to click the setup environment. The setup environment will install the MAFFT. Also, it will install the IQ tree program to this notebook. It may take almost 1 minute. So for the reason, I will uh, pause the session. Now it is ready. It took almost 30 seconds. Okay, clear the selected output. That is not necessary, but actually I do it. Okay, so now we are going to upload our data file. So click here. You will see this choose file option. Choose. Now my data is in on the desktop. Okay, and data. Here's this one. Okay. Now the data is uploaded. Once you click here, this option, now you can see IQ tree which is installed and rbcl underscore gene dot txt. This is actually a plant-based genes and they are using in the DNA barcoding. Okay, so this cell will verify like our data is successfully uploaded or not. So just click here. If you see this kind of thing, it means the notebook is quite handling well your data, and that's it. Okay. Now, there are some important read. I will train you on two strategies. To the first one, I call it strategy one, and to the second, I call it strategy two. Okay. What is strategy one? In strategy one, we will align our data set and we will also generate a kind of simpler kind of tree. We call it guided tree. Okay. In strategy two, we will use IQ tree and we will generate a very professional kind of tree and we will perform a professional kind of analysis. What actually is guided tree and what is the difference between the guided tree and the tree we will generate in the strategy 2? The guided tree actually do not read or do not entertain all those models, all those evolutionary models. We call it algorithms or models like JT, GTR, gamma, etc., etc. But it just randomly generated from the alignment, input alignment. So that is not recommended for the publication quality or for the publication analysis. So that's why, but so far we will perform both of them. Also, the strategy one can be okay for quick visualization and for smaller data set, but not for the publication quality images or publication quality analysis. What are the key differences between the MAFFT tree and the IQ tree? The MAFFT tree will be generated in strategy one, as I told you, and the IQ tree will be generated in strategy 2. So for the MAFT is quick for alignment and it's for quick visualization, but the IQ tree will give you higher accuracy. MAFT just going for the sequence similarities, while the IQ tree are using the maximum likelihood and other advanced model. The MAFT is, you will feel if your data is bigger, you will feel that the MAFT is slightly faster because they just perform the pairwise similarity. It could be a bit slower than the MAFT, but that doesn't matter. The MAFT accuracy is just like random, IQ tree is highly accurate. MAFT cannot provide you the branch support. If you are interested in branch support, then or the bootstrapping and all those things, then uh, you have to use strategy 2. Okay. MAFFT cannot be used for evolutionary model, and this can be used, and the MAFFT tree is rough. This the IQ tree is not rough or kind of tree size. Okay, and best for journal visualization, and the IQ tree is best for publication quality analysis. Okay, and okay, now you need also to know, okay, first we are going to perform the strategy one. So what kind of new file it will generate here. So let's see. First, it will align our sequences and then it will generate a tree. Okay, so you will see some new files will pop up here. Okay, the first file is rbclgene.txt.tree. So it is actually the tree file. If you want to open it, just download, and it will be in the download folder. So click here, open, and let me go to the download folder. Yes, here. So you can see it's a kind of tree, but it do not contains any branch support. It's kind of rough tree generated by strategy one. Okay. So okay, another thing, it generates aligned dot fas dot fas. So alignment.fas is actually the aligned file. If you want to store it, just download, and now we can see like whether it's correctly aligned or not. So oh wait, probably we made some mistake. And okay, so I spotted there was a smaller error. When you click here, you can see the input file and rbcl underscore gene dot txt. Before it was dot fas. So we have uploaded a txt file. So that is why I fixed it. And now I hope this file will be okay and can be opened also. So let's see. Yes, we have 19 sequences and uh, about you can see the gap. So it means the data is aligned. Okay, so don't confuse yourself. Uh, if you upload the file here, let me clear it. If the name of your file is abc.txt, so what you have to do here, click here, wait, strategy one, click show code, and then, for example, this is your file, you upload it, click here, copy path, and then do this, Control V, and now run it because it actually read the uploaded file. So you have to change this information because the information I stored already are not kind of the the names I use are those which I selected, but I don't know what name of file you will upload it. So I hope this step is clear now. Okay, so we successfully had generated the tree file. So now I'm going to delete these files because in next step we are going to generate so many other files. So that's why I need to delete it. Okay. Now we are proceeding towards the strategy two. But for the strategy two, you need to understand few things because it's kind of professional strategy. So you must have some basic information. The IQ tree, it uses different evolutionary models. We call it JC or Ju Cantor, K80, HKY, SYM, or symmetrical JTR, TNN, blah blah. You can read all this. If you want to use a specific model in your analysis, so you will just indicate the JC or K80 or HKY, whatever. Also, the IQ tree supports models for the heterogeneity test. For example, plus I option will proportion of invariant sites, plus G option it means gamma distribution. You need to read if you are interested these things, and if you are following some paper and if they have used some specific model mentioned here, some specific heterogeneity test, so you can also apply these things. Okay. The third thing, if you want to use these two things in combination, for example, you want the gamma distribution with the GTR, then it will be GTR plus G, or you can read this, it's not that difficult. Finally, if you are not so expert in bioinformatics stuff and you want me to do something that the program itself detects the best model for your data, then we will use this option MFT. The MFT option will automatically test all these models, all these combination on your dataset, and finally it will tell you what are the best models or the heterogeneity test best models, or what are the best combination for your data, and automatically they will consider it. So now click here. So if you see here, we have this code include this is the function we will run. This is the input data, which is already the align.fast is already generated from the strategy 1. Okay, now I'm using MFT. It means I'm asking the program to decide which is the better combination or which is the best model for my set of sequences. But once again, let me tell you, because sometimes students are following some other papers and they want to reproduce the same methodology. So in that case, for example, if you are interested in the, let's say you are interested in the HKY model with the combination of plus I or the proportion of invariable side, then you have to do like this HKY plus I. That's it. But in this case, I will ask the program to find out what is the best model and the best combination. But if you are also interested in the bootstrap calculation, so by default, I put it 1000. You can increase or decrease this data, this value. NT means nucleotide and R2 means, like I asked it, like you can run your auto function and find out which one is the best model for our dataset, and then give me a complete record or a complete analysis. Okay, so I'm going to run this cell. Let's see what it says. Okay, it says no such files are directory, probably because I did not properly install. Let me check it. Okay, I rerun the first code, and because by mistake I thought we have deleted this folder. So let me fix this issue. I will save this version so you will not have this issue. Don't worry. Okay. Now the IQ tree is running, and you can see it calculates or it's entertaining all the models and all those things. Okay. The successful execution will give you the Sunday, April 20 and the time. Okay. Now, just understand this part now. So many files are generated here. You can see so many. Now, but there are typically five files which are important for you to understand. The first file, tree.output.tree file. So let me check, do we have .tree file or not? This one, align.fast.tree file. Okay. So if you are not aware of the computer language, so I would suggest to trace the file by this name dot with the extension name. So .tree file. So let's say download. Now we have this file in the download, and let's open one what it says. Okay, this is actually the Nivik format .tree file, and your tree is generated with all branch supports and each and everything, but we will explore a bit later. Now, the second file is the full model information output tree, and it will be ended with the IQ3. Do we have this file? Yes. Click here, download. Just analyze this file. Okay, it tells you about each and everything. Okay, about the citations, like if you are using this program, what to cite, about the sequence alignment, like how many positions in our data we had, 1428 positions, how many constant sites and how many invariable sites, and number of parsimonious sites and number of distinct patterns. Okay, this fit model according to TIMFI, it means they have tested all these models you can see, and these are the model scores. If you want to use this table in your thesis or publication or in supplementary material, you can take it from here. Now, also, it tells you about the rate of substitution parameters under different models, so you can read this. And it gives you a tree here, but don't consider this tree. Then they give you the Nivik format tree for this tree, information for this tree, and then they give you the final tree, which they call it consensus tree, this one. Okay, so but you have already this tree downloaded, so just close it. Why I wanted to show you this file? Because this file will help you in writing of your thesis and papers, and you will find out these information. This is the log file, you can see it's here, and this is the next file is the model.gz. It should be somewhere here. It, if you download it here, it's the compressed, it will contain the compressed data. You can unzip it or uncompress it, and you will have all this data if you need, but still we downloaded these file individually, so I think we don't need it. Okay, this is the ml-dist file, because in this analysis we use almost 19 sequences. So that means that these nine, what is, if you are interested to extract the pairwise distance matrix, like sequence number one, how much far or how much near it is to another sequence two, three, four, five. So this is the information of the distance matrix. Uh, you can also plot the distance matrix information, although I did not include it here in this notebook, but you can find out so many things, so many server to plot that this matrix information. Okay, finally, which should you choose, or what should you choose? Because I generated two trees for you in these analysis. The first one was all about the simplest tree generated in strategy one, which later on I deleted. And I also suggest you to delete that because that tree is not so accurate. Uh, that was with the flag or with the function tree out we generated in strategy one. But as I deleted, I would also suggest you to delete it. Okay, for professional and kind of more sophisticated analysis, I would suggest to carry on for strategy two and generate a very good tree or whatever. Okay, now we have the downloaded tree here. This the tree file. Now, if you don't know how to visualize it, so you can open it in Mega, you can open it in BioEdit, but I would suggest you to, if you can, just open this iTool, then upload your tree. Let me upload this tree. It should be in the download folder, this one, tree file. Okay, upload. And after upload, you can have this kind of tree with branch length. You can do different things. I have already covered another video with this probably, but it's not that difficult. It has four tabs: basic, advanced, and export. The dataset is highly professional tab, so I would not recommend if you are a beginner or a basic user. So with the basic things, you can just explore what kind of tree you want to visualize it. This is actually the tree of our dataset. But if you want to uh, ignore the branch lengths or to use the branch lengths, you want to use this kind of tree or this kind of tree or this slanted tree or height. If you are interested to display, if you are interested to change the display font from here, alignment right, alignment left, rotation, blah blah blah. Now in the second tab, advanced, if you want to indicate, let me check it to rectangular. Yes. If you want to indicate the branch lengths from in advanced option, so you can see all the branch lengths are indicated, or if you want to hide it. Okay, if you are interested to indicate the bootstrap confidence, as we have uh, generated the 1000 bootstrapping, so click this display. Now you can see for all nodes some specific circles are here. The bigger the circle means the higher the confidence. Okay, so this is all if you want to select some bootstrap range like below than this range, do not show me the bootstrap values. You can do like here. If you want to change this bootstrapping visualization things, the sign, the steric, or this program gives you only these few options. Or if you are interested to change into some different colors, let's say this one, then you can see here. And if you are interested in some border weight, then some you can see uh, the borders are changing. So that's up to you. And finally, you can export your tree here in your favorable or in your favorite format. Okay, I hope from now on you will be able to generate a very good um, phylogenetic trees and to perform phylogenetic analysis on your data set. If you're liking my video, please thumbs up, subscribe, and share with your students, colleagues, and fellows. And I made this video after a long time. So currently, I'm not feeling so well, so that's why. But I hope you will love this tutorial. And for next tutorial, I will come up with some how to deal protein data, how to tweak with different models and dealing with protein data. Thank you so much from now. Take care. Bye bye.